Welcome to the Theoretical Neuroscience Podcast. I am the host, Gaute Einvoll. The field of computational neuroscience encompasses many different types of modeling. Broadly speaking, most activities can be classified as either inverse modeling or forward modeling. In inverse modeling, one starts with neural data and then uses statistical analysis techniques to extract patterns to help in the interpretation of the data in terms of the underlying neurobiology. In forward modeling, one instead builds models based on what is known about neurobiology, and then compute what the models predict and compare predictions with neural data. The most famous forward model is probably Hodgkin and Huxley's model from the early 1950s for the generation and propagation of action potential in squid axons. Here, the key building blocks of the model are particular sodium and potassium ion channels embedded in the axon membrane. Since this pioneering work, most forward modeling in computational neuroscience has been at the level of neurons, often focusing on how neurons generate action potentials based on inputs, or at the level of neural networks, focusing on collective firing properties of many neurons connected together. But there is a crucial level below, that is inside the neurons. Here one finds complicated signaling pathways where numerous molecule species interact with each other and form new molecules in cascades of chemical reactions. An example is the complicated signaling pathways that, following an influx of calcium ions into the postsynaptic side of a synapse, lead to long-term strengthening or weakening of the synapse. In neuronal network models, one typically includes such synaptic plasticity effects in simple phenomenological models, describing how the synaptic weights change depending, for example, on the times of presynaptic and postsynaptic spikes, such as in the simplest spike timing dependent plasticity rules. However, if one wants to understand what is going on a deeper level, one must explore models formulated in terms of concentrations of the numerous molecular species involved in the process. This typically involves solving a large set of interconnected ordinary differential equations, one for each of the molecular species involved. It is thus quite a bit more mathematically complicated than modeling of the dynamics of one variable, the membrane potential, in the case of electric neuronal dynamics. Today's guest, Avrama Blackwell, is one of the leading experts on this kind of modeling. She just moved to Iowa University in US, where she is a professor at the Department of Biomedical Engineering, leading the computational and experimental neuroplasticity laboratory there. In the podcast, we talk about the different approaches used when modeling signal pathways and the kind of experiments that is done to determine model parameters and test model predictions. Calcium ions are not only involved in determining the electric properties of neurons, they are also key signaling molecules inside the neurons, involved in controlling numerous intracellular processes, which we discuss. In the podcast, Avrama points out that calcium ions may cluster in so-called nanodomains, possibly allowing for highly local computations in small segments of the dendrites. So the spatial scale of computations with calcium may be much smaller than for electrical computations. As for all episodes, information about the episode and relevant links can be found at the webpage theoreticalneuroscience.no. If you want to support the podcast, go to patreon.com/theoretical neuroscience podcast and choose a membership of your liking, for example, at the cost of $1 per month. And as a member, you will get access to a video version of the podcast recordings as well as episode transcripts. And note that there is a 7-day free trial period when you sign up. I also have a YouTube channel called Theoretical Neuroscience Podcast where you can watch the videos as well. I will be most grateful if you become a Patreon supporter to help me fund the production of the podcast, even if you're not too interested in looking at the videos. But I also appreciate all the other listeners, and I hope that all of you can help spread the word about the podcast to other people in the community, on social media or otherwise. Okay, here is Avrama. So, welcome to the podcast, Avrama Blackwell.
Thank you. I'm delighted to be here.
So, so this topic of today, signaling pathways, is sort of like a minority activity in computational neuroscience, I think. So maybe we should just start by Well, maybe you can just explain what is meant by a signaling pathway.
Right. So a signaling pathway is basically a cascade of biochemical reactions which lead from a synaptic input to different cell processes, which are really critical for all parts of the cell. Um, what many people mean by signaling pathways is signaling to the nucleus. And
Mhm.
indeed, it is relevant for the brain because we know that functions like synaptic plasticity absolutely require gene transcription
Yeah.
and
So
protein
gene transcription,
synthesis.
that, yeah, exactly. Just so gene transcription, that means that you sort of transcribe genes into proteins, or is that what you mean?
It means so gene transcription is you activate the gene so that you get mRNA, right? The
Mhm.
message. And then the protein synthesis
Mhm.
is you go from the mRNA to the protein synthesis. And those are two separate functions because in
Mhm.
some case you can stop one and the
Mhm.
other still occurs and
Okay.
vice versa.
Okay. Okay. I think I interrupted you. So you said that that's one important signaling pathway, obviously,
Right.
to make the appropriate amount of ion channels, for example.
Yes. And but most of us that study signaling pathways, we don't get all the way to the nucleus. We're looking at more local events.
Mhm.
So we know that signaling pathways lead to phosphorylation of
Mhm.
existing proteins, which either
So
change
what
their
is
activity.
phosphorylation? So okay, so just go through the
Okay,
molecular, like, whatever card part,
right.
molecular parts first, molecular players. So I guess you have proteins and
So you have proteins, and they have a particular three-dimensional conformation,
mhm,
which determines their function.
mhm,
Right? So, in other words, an ion channel will open and close, but perhaps the amount it's open or the amount of ion flux through the channel when it's
mhm,
open is controlled by its structure.
mhm.
So, a phosphate group is this rather large molecule of a phosphate and a couple of oxygens um, that take up space and modify how the conformation of the protein.
So that's sort of like a group that attaches from the inside on the ion channel.
It's a group that will attach. So the protein consists of a string of amino acids.
Mhm,
And interestingly, there's only a few amino acids which are really phosphorylated. That's
mhm,
serine, threonine, and
mhm.
tyrosine.
Okay.
Okay. And so a kinase is a molecule that will take an ATP, an energy
Mm,
molecule,
mm.
take off a phosphate and put it onto the protein on one of these amino acids.
Okay.
And what's really interesting is that
Yeah,
the
because just like
kinases.
proteins are made of amino acids, so you have
Mhm.
some amino acids then that are sort of like receptible or can are able to accept these phosphate groups.
Right,
Mm.
right.
So, mm.
And when you put that phosphate group on,
Yeah.
it changes the shape of the protein, which can change its function.
Okay, so these must be then be amino acids which are at the sort of like at the entry point or the inside of the ion channel, maybe then, so that the phosphate group can get attached.
That's a really good point. So
Yeah.
not in fact, it not only has to be on the cytosolic side or the
Mm.
inside, but
Yeah.
it has to be accessible. So you think of these really long strings of amino acids and they're coiled in such a way
Mm.
that there might be a whole lot of serines and threonines, for example, but there might only be one or two that the kinase actually has access to. So a lot of neuroscientists they identify which of the amino acids are being phosphorylated,
Mm.
say by some kinases
Mm.
in the course of cell function
Mm.
or
I
activity.
see. Yeah, so this is sort of like an example of like a signaling pathway where this new molecule, the phosphate, comes into play, and typically sort of the regular. I mean, for when you're doing computational neuroscience at the cellular level, often the only thing you worry about is the membrane potential. So it's essentially that's the main dynamical variable. But here essentially you have many players. You have many different molecules that interact with each other inside the cell, and then that gives eventually this phosphorylation, for example, which is this example pathway.
That's correct. And even without the phosphorylation, there are some ion channels which are sensitive to second messengers. So a
Mm.
signaling pathway, as I said, is a cascade of biochemical reactions.
So
So for
cascade, you mean
example,
that it's like
a
many sort of like
sequence,
a
many
sequence?
of them.
Mm.
That's
Yeah.
right. So you have calcium flows in and it might bind to a molecule, which then binds to another molecule, which
Mm.
then produces yet another molecule. Or in the case of the more commonly are G protein coupled receptors.
Mm.
So these molecules are really interesting because the set of these molecules is huge.
Mm.
Okay.
So what is
And
G? Okay, so tell more about what the G protein. That's
right.
like that's I've heard I hear that many times. What is it?
So let me explain first of all why they're so important because
Okay.
something like 50% of our psychoactive drugs, our
Mm.
therapeutic drugs, are targeting these molecules.
Mm.
So this is a receptor. So it's a protein,
Mm.
it's in the membrane, and it can bind to a molecule that's released presynaptically.
Yeah.
Now, we often in neuroscience computational neuroscience, we think of the synaptic channels like AMPA receptor, AMPA type and NMDA type glutamate receptor channels.
Yep.
So the difference between the G protein coupled receptor and these what are collectively called ionotropic
Mm.
channels is that there's no channel involved. So AMPA, a glutamate binds, the channel opens, uh, ions flow in. With a G protein coupled receptor, the abbreviated GPCR,
Mm.
this molecule binds, say, glutamate. There's no channel that directly opens. Instead, that GPCR that's bound to its ligand is an enzyme,
Uh-huh.
and it catalyzes the activation of, not surprisingly, a G protein.
Mm. Yeah, I see.
All
Yeah.
right, a G protein is short for a GTP
Mm.
binding
Okay.
protein.
Yeah.
Okay. So
Mm.
this molecule gets active and then it binds to another enzyme and activates it, which then produces typically a diffusible second messenger such as cyclic AMP.
Mm. I
Now,
see.
the cyclic AMP could do one of two things, and one of which there are certain channels which actually their activity depends on the concentration of cyclic AMP.
Mm. Yeah.
So it just binds and it changes the activity.
Yeah. So this
So
second
even
message,
without kinase, you can get a modulation of ion channels.
so this metabotropic glutamate receptors like GABA B. So that's one of these things, right?
So, G protein coupled receptors is a big class.
Mm.
Then there's a metabotropic glutamate receptor, like mGluR. There are indeed metabotropic GABA receptors, so that would be the GABA B.
Okay. Yeah. Oh, exactly. Yeah, of course. Yeah, that's the. Anyway, so we're already into the the zoo of molecules. So,
Yes,
of course, which we typically sort of is sort of hidden under the carpet when we sort of model at the neuron level, right?
that's right.
So for example, one very important thing is synaptic. I mean, synaptic function and synaptic plasticity. So we'll come
Hmm.
back to that because that's also obviously this thing that is weights of the synapse change over time, which we then have all these sort of like phenomenological phenomenological models for, which are typically used then in say network simulations. But there
Right.
are also of course there's a whole yeah, it's a whole network of it was a big signaling pathway under the hood there, right? So which is
Yes.
also interesting. But first, I think, I mean, if you sort of look at the how, if you, of course, when you want to make models, you need experimental data. So
Yes.
what kind of, so here you need to measure molecule concentrations or something like that, right?
So there's a lot of different types of measurements.
Mm.
Some of the most basic ones, of which there's more high throughput data, is saying which molecule is binding to another molecule.
Mm.
So that's known as protein protein interactions.
Yeah.
So I know that cyclic AMP binds to protein kinase A. So that would be one type of piece of information.
Mm.
Then we do
So
indeed,
where do
we'd
you get
love
that information from then? Then you get that from,
um,
so which protein binds
so
to which
so
protein?
a lot of these experiments are biochemical experiments. There's a technique called yeast to hybrid, which is a high throughput technique to say what molecules just bind to other molecules. But then you're verifying each of those reactions, perhaps by some other biochemical assay. Um,
Mm. I see.
other
So yeah.
Otherwise, I think like a pull down assay, like you have your tissue, you have an antibody to
Mm.
one of your proteins, and then you pull it out, and then you can blot for your other protein of interest that you think is binding to the first one.
I see. Yeah. So there's a whole, yeah, exactly. So you have all these traditional, obviously biochemical techniques, or
Right.
traditional. I don't know. Yeah, probably old ones, the new ones. So the techniques are probably improving, and then you also have some optical techniques, right? To
Yeah, so what we didn't talk about was the kinetic rates. So we want to know, well, this molecule binds, but
mm,
how fast does it bind?
mm,
Or once the protein is active, if it's an enzyme, how fast does it produce product? And so once again, there are those traditional techniques, um, biochemical kinetic assays, and the newer techniques. Their advantage is you can measure the activity over much shorter time frames to see a really, because typically the initial rate is higher. But
mhm,
then, um, unfortunately, those techniques are often done by pulling the enzymes out of their native situation. And some of them will work better if they are in a membrane, for example. Or if you purify it too much, it may not work well. If it's not very pure, then you think it's working slower because you don't have as much enzyme. So
mm.
the newer techniques are imaging techniques, which don't actually provide you with single rate constants, but are actually measuring the dynamics of the molecule changing concentration in situ at a great time frame that's
So in
relevant for
situ
neuroscience.
means in vivo. Is that
In
no in or
cells,
in
yeah.
cells? Yeah.
I I mean, you're not some of the work is done in vivo, but
Yeah.
most of it
In
is not
vitro.
done
Yeah.
in vivo. It's
Yeah.
really done in, say, cell cultures or brain slices.
Yeah.
And so what I mean is the molecules themselves are in their native environment with membranes and stuff,
Yeah.
even if it's not
Mm.
actually in vivo.
So, so of course this is must be a big challenge because there's so many in this signaling pathways that one are interested in. There are often, I see some of these models have like hundreds of different, I mean hundreds even of different molecular species, right? And ideally you would like to have
Yes.
measurements of the concentrations of all these, right?
Yes, and that is true, but I don't know if it's really that much more difficult than multi-compartmental ion channel modeling, where
Well,
you have tens of ion channels, each of which could have dozens of parameters.
mm,
And you could even have, again, the ion channel might be expressed throughout the dendrite, but the phosphorylation state,
mm,
getting back to signaling pathways,
yeah,
could be different out in distal dendrites, which means it acts differently.
yeah,
So I think that
mm,
the the challenge is a little bit more,
mm. Because that's
but
something I mean, if you do sort of like multi-compartmental modeling, then typically you assume some ion channel densities, different ion channels and densities of those, and but you're saying that because this phosphorylation, that's typically not included in the model. So that's, you say that is like maybe like a major point that we tend to ignore and maybe at risk.
well, we not only ignore that, but honestly, it's very difficult to get good kinetics of our ion channels. And so
Mm,
often they might only be measured in one brain region
mhm,
or two brain regions, but not the brain region you want. For example,
mm,
the sodium channel, it is so fast that it's actually very difficult to do a voltage clamp. So somebody might have voltage clamped it in a rat
mhm,
neuron and you're using that for a mouse neuron, or
mm.
somebody did it in the hippocampus and you're doing it for a brainstem neuron. So
Yeah,
I'm not saying that it's easy finding the rate constants. I'm
yeah,
just saying that
uh,
people minimize that the fact that it's also very difficult to do that for
Yeah.
ion channel models.
But this thing of sort of like people also do, they sort of take this, they transcribe these particular ion channels like, as in like some kind of oocytes, some empty cells, right? And
Yes.
then so that seems like a cleaner thing, and maybe you could also measure it there with and without phosphorylation, or if that's sort of
Well, actually,
it's hard
in
maybe.
that case, the issue is we now know there are a lot of auxiliary subunits um, that change the activity of the channels. So when you express, say, typically the pore forming subunit, also
Mm.
called the alpha subunit,
Yeah.
um, it probably doesn't have its auxiliary subunit. So the activity you
So what is
measure,
auxiliary subunit? Something that is like hanging outside the tunnel?
yeah, it's associated with the channel.
Yeah.
So in some of the ion channels are called the beta subunits.
Mm.
In potassium channels, there's something called K chips. So yeah, so either they don't have auxiliary subunits, they have a different phosphorylation state that they would in your brain region of interest, or there are other factors. Again, for HCN channels, which are also called by modelers H channels, those are modulated by cyclic AMP. And even in ion channel models, they often ignore the effect that cyclic AMP concentration might vary and therefore change the activity.
Yeah. Mm. Hmm.
And
Yeah.
there's several calcium dependent potassium channels where
Yeah.
the calcium,
Yeah. So
is
there's a lot of complexity at all levels, not only below the hood where you are working inside the cell.
yes.
So, but sort of, I mean, there's something. I mean, like if there's also this field called systems biology. So, what
Yes.
is systems biology? The way I mean, as you understand it.
Okay, I'm glad. Let's qualify as I understand it.
Hmm.
It's understanding these biological systems as a system. And so there's a large modeling component to that, which indeed tries to understand a lot of these pathways, not just the signaling pathways, but even metabolic pathways
Mm.
and gene transcription networks. And so there's a lot of these newer high throughput biology techniques, such as transcriptomics, and again, these protein protein interaction techniques um, to understand, you know, large quantities of data. And then there's different approaches to modeling that,
Mm.
um, ranging from the ones that just look at what molecules are interacting with each other,
Mm.
regardless of concentration or rate constants, and then other models which try to un- and they don't even look at the dynamics, meaning how
Mm.
things change over time. But then there are other techniques which try to look at how molecules change over time without the need for knowing either rate constants or protein concentrations.
Hmm. Yeah. So yeah. So but so I mean, what the signaling? I mean, systems biology. If I mean, if you're completely new to biology, you would think that it's actually in practice restricted to what goes on more or less inside the cell, the way it has been used. A little bit like bioinformatics, which also has a like a narrower meaning inside the field than what you maybe would expect from if you just, I mean, applied of informatics to biological questions. That's much more than just what goes on inside cells, right? So it's a little bit systems biology is a little bit of a we do also systems neuroscience, and then we mean maybe more at the network level.
Yes, but I would hesitate to say that systems biology only looks at what happens in the cell,
Okay.
because they do look at, again, ligands, right?
Mm.
Extracellular, not neurotransmitters, but
Mm.
other molecules released by cells. And in some cases, people are, for example, looking at when they study cancer, the problem with cancer, one of the problems is that cells lose the ability to recognize each other, to sense that we're getting crowded. And so in that case, they are applying systems biology to understand the issue of networks of cells, though not how we define networks, but
Mm,
you might say clusters of cells,
I see.
and why they're not recognizing those contacts.
So exactly. So it's going a little bit beyond a single cell. That's what's saying that
It
it's
could,
yeah.
yeah, in some fields.
But I mean, like ecosystems in the jungle is nothing. That's okay. Could also be
That
systems that
right, that probably
would
I
not.
mean, technically that's
Yeah,
systems biology,
yeah,
though to be fair,
yeah.
you could look at any field and look at the nomenclature
Mm.
and have a quarrel with it.
No, yeah, no, no. So I don't have a problem with it. I think also with bioinformatics and it's also like a term because that's where the I think the activity where you could actually use techniques from I mean computer science and so on. So then it
Right.
just grew into that. So it's I have no grudge against the people or the bioinformaticians or system biologists. Just that it's. I remember I was a little bit confused by it myself at some point. But
I
yeah,
see.
it was sort of more when people talked about systems biology, they typically talked about what was going in on inside cell, and I was a little bit, which is
I see.
clearly deserves to be called systems biology, but you could also, in some sense,
That's true. So, I mean, system is a very vague term,
mm,
right? So, as you said, in neuroscience, we think of a system as not even being a small network of cells, but being a really much larger
mm,
population. Whereas in systems biology, um, the spatial scale is much smaller, and yet the system is equally large
mm.
because these networks are huge because there's so many different interacting
That's true. so,
proteins.
so in
S-
terms of this,
Um,
the size of the models become really big because
right,
there's so
but
many
you can
actors.
also but think about power systems in the US, right?
Yeah. Mm.
Those people might say you're not studying a system, you're only studying a single brain.
Yeah,
Whereas the power system might be
yeah,
studying the entire United States power
absolutely,
distribution network.
absolutely. Yeah.
So,
So now, I mean, I'm sort of following a little bit your. I think you wrote a very nice review article together with Jeanette Helgren Kotaleski. I think it was like I'll certainly make a link to it on the homepage of the episode about sort of like the different techniques for doing this kind of signal pathway
uh-huh.
modeling. And then and then you start with this thing that you sort of touched on, this thing of this simplest modeling is sort of like graph theory or the kind of sort of the simplest model where you just sort of look at sort of what kind of say proteins that sort of come together and produce new proteins, or how sort of how the different concentrations come about.
So graph theory doesn't even look at concentrations. So I don't want to say theory is simple. That perhaps would be an injustice, but only in the sense that you don't need constant molecule concentrations and you don't need rate constants at all. You're really looking at these networks of interacting molecules. So you start with, again, this protein protein interaction. You know that protein A interacts with protein B, and maybe protein B then interacts with protein C. And you have large numbers of these um, interactions that you know of.
Mm,
And you're looking at you can create a graph. What that means is each protein is a node and every interaction is an edge.
mm. And that's
Um,
sort of like the connection between nodes, right?
yes, the connections between nodes. And so think about the classic one um, graph theory is applied to is airlines.
Mm,
Think about all the different airports, and some of them are tiny and have only direct flights to a few other airports, and some of them and that would be where I live now, Cedar Rapids Airport.
okay.
And then you think about Chicago O'Hare Airport,
Mm,
which has connections to so many different places, and you can draw that as a graph, so it's easier because it's two dimension. And so you put all the airports and then you draw lines, and everyone who's flown has probably
mm,
taken the trouble to
mm.
look at those maps in the back
Yeah,
of the
yeah,
those magazines that they used to give us.
yeah.
And so you can do the same thing with proteins though, and you discover that there are a small number of highly connected nodes
Mm,
and a large number of less well connected nodes.
mm.
And so, for example, let's say you're trying to understand something that's very complex, like intellectual disability, for which a lot of work has been done identifying mutations. And it turns out that there's a lot of different mutations that occur very infrequently. And so that would make that disease really difficult to understand. How can we have all these different mutations that produce something that looks similar in phenotype, meaning in behavior of the person?
Mm.
And so, what graph theory can show us is that many of these mutations might be connected to, say, a hub protein, and the hub is the one that's highly connected.
Yeah.
And so, maybe the reason they look different is because all of these mutations or proteins that occur infrequently are all feeding into this one very critical protein. And
Mm.
therefore, what you're seeing in terms of the phenotype is the dysfunction of that hub protein.
Yeah.
And that's what graph theory can show us.
Mm. You also mentioned in this that that you can also find this what's it called a phosphoprotein network, and you mentioned that what is it? It's a phosphoprotein network of the synapse at like 1,500 proteins. That's my goodness.
Yes.
Yeah.
So we tend to think of these, again, systems biology. Let's just look at all the proteins in a neuron
Mm.
and let's ignore the location of the proteins. So one drawback of the graph of graph, not really graph theory, but just general protein interactions, is we ignore the fact that, first of all, different proteins could be in different cells. And if they're in the same cell, they could be in different parts of the cell. And so, to really try and understand synaptic transmission and synaptic plasticity, this group that I referenced, they isolated synapses and then applied this sort of, let's look at all the proteins and look at the protein interactions in the synapse.
Mm. So that was
And
like a presynaptic or postsynaptic or both, maybe, or
I would have to check
yeah,
the
that's
paper
not so
there.
important. It's
Yeah,
it's probably a bunch
there's
in both.
different preparations,
Yeah,
and I
that's
don't
fine.
remember
No, that's
which
fine.
one they used.
Mhm. So then, yeah, because I just read like 1,500 proteins. So that's a lot of proteins even inside the cell. So and then you after these sort of like more like static networks that you sort of have with this graph theory and like in looking at the topologies and so on, network topologies. Then the next step, of course, trying to do some dynamic modeling. I mean, try to model the dynamics. And a key assumption there is that things are well mixed typically. That's typically what does that
Yes.
mean?
So well mixed means that the when you have a, let's say you have a container, and let's
Mm.
just talk about the cell body, and we'll just approximate it as a sphere. So the concentration of the molecules is the same in all parts of that cell.
Mhm.
And the implication is that every protein has an equal chance of interacting with every other protein
Mhm.
if such an interaction can occur.
Yeah.
And that simplifies the mathematics for modeling that system tremendously. Also simplifies the knowledge you have to know about the system.
Mm. Yeah. And the simplest or the simplest everything is relative, I guess. It's like these Boolean networks of such. So what are those?
Yeah,
What are those?
so that's a really cool technique and allows you to do these models without knowing the rate constants or the concentration of the molecules, which again is a limiting is factor because it's more difficult to find. So you develop these rules which says if you have molecule A, so every molecule is represented as a single number. It's either a 0 or a 1, meaning it's either not present
Mhm.
or inactive,
Mhm.
or it is present or is active. Okay?
Mhm.
And so then your rules start to be, well, if molecule A and molecule B are both 1, then
Mhm.
molecule C at the next time step will become the value 1.
Mhm. So that's like
Okay?
an and gate. So almost like
It
an
is
and
and,
operator.
ors,
Yeah.
exactly,
Mhm.
and
Kind of.
nots.
Yeah. Exactly. Yeah.
So but with that logic, so instead of saying molecule A and B will react according to this time scale, they just either react or they don't. And instead of saying molecule B, now we have a little bit or we have a lot or we have really a lot, it's either present or absent. And so and you there's different ways you can run the networks. They talk about synchronous or asynchronous, and often though you're looking for the network. You start at different initial conditions and then you're trying to see where does the network end up. Is there a stable solution?
Huh.
And maybe the how you run it will determine how long it takes to reach that equilibrium. And there's a newer variant where you can, instead of making it you either react or not, you can give it a probability of reacting.
Hmm. Yeah.
Um, but it's still, and they've made some predictions with that, which I find quite remarkable. Predictions meaning that they got experimentally tested and then they were correct.
Hmm.
But I don't think you're going to be able to see a time scale of how when things happen.
Hmm.
Which I think the time scale for neuroscience is so critical because our signaling events are so brief.
Yeah.
Say
I
if
think
you're
what I
looking at
mean
a
is.
metabolic network
Mm,
where the time scale is much slower, that may not be as important.
mm. Yeah. But I guess there are time scales in like learning is also can be quite slow in neuroscience. I mean, so obviously the fastest time scales are milliseconds, or well, the
Well,
spikes are milliseconds, right?
that's a really good point. Learning is a funny thing because certainly it can happen really fast, right? Think one
Mm.
shot conditioning,
Yeah.
right? If you go to a new restaurant and then you
Mm.
get sick that night,
Uh,
you will never, never go back to that restaurant.
no,
Okay. Um,
but, but it's not a millisecond response until
well,
unless
it's not milliseconds, but it's pretty fast.
yeah, it's pretty
And
fast.
there are other situations where they show,
I see.
using other stimuli, right? Taking into the lab where it occurs pretty fast. You're right. It's not millisecond,
Mm.
but the events themselves which lead to the memory storage
Yeah.
can be certainly seconds to
Mm.
minutes, which is still very fast. We don't necessarily see the memory expressed for a longer period of time, but the events that trigger that memory storage can actually be quite fast.
Oh. And then I guess the next step is then try to sort of model. I mean, model the dynamics of concentrations, and there, I
Yes.
guess, with this mass
Right.
action law. That's
Right.
actually one of the first. I think it's the only law which was sort of well is attributed to Norwegians, and in terms of like a
Really?
semi-natural like Gullberg Våge. There was like a chemist and a mathematician in the late 19th century. So that's
Oh,
so that's well, it's a pretty
fascinating.
important law. Yeah. Anyway, so that's a little bit of patriotism here. Anyway, but what is that seems to be the then you end up with this a bunch of I mean in the well mixed case you end up with a bunch of differential equations, right?
That's correct. So there's a pretty well established theory. Actually, what I learned about it first was called biochemical kinetics.
Mm,
So that if you have two reactants, say, that produce a product and you know the forward and backward rate constants, then it's very straightforward to write that ordinary differential equation. And you can do that for a whole bunch of different reactions.
mm,
And so I like to say for every set of arrows you have one differential equation.
mm. Yeah.
And so that's why if you have a whole series or a system of biochemical reactions, you have a system of ordinary differential equations.
Mm. And I guess one sort of one thing that you have worked on is this, and I also we have sort of worked together a little bit with sort of with Tumo Meikki
Yeah.
Maitonen, a postdoc working with us. So on this use of this kind of working on these large models with a bunch of molecular species for this account trying to account for the synaptic plasticity like long-term LTP and LTD.
Yes.
So what can you tell about this process? It seems like is it right to say that that it sort of starts with it always starts with calcium. Oh, we have a cat coming here. Hello.
Yes. So, so we have the inputs to the model, and a major input is calcium. Calcium is
Mm,
required for certainly every form of synaptic plasticity that I'm aware of.
yeah.
But calcium is not just an ion with charge, it is a second messenger itself. So it
Mhm,
binds to a lot of different molecules. Some of them we're going to call buffers, meaning they're molecules that after the calcium binds doesn't really do anything. It just sits
mhm,
there and prevents the calcium reacting. But it also binds to something called calmodulin, which is a calcium buffer. But calmodulin bound to calcium can now bind to a whole bunch of different downstream molecules and activate them and set off these signaling pathways
yeah.
that then interact with some of these other ligand inputs to the GPCRs.
Exactly. And then you can, depending on what of these signaling pathways gets activated, you can get both sort of LTP, long term potentiation. It's like a strengthening of the synapse, or the opposite,
Correct.
depression. So these are quite challenging models. I mean, I mean, it's like several. I mean, I think with the largest model that Tuomo made, that was like a few hundred species or something. But there are also simpler
Yes.
ones, and then you have to run them for a long time, I guess, because it's the induction of plasticity is a quite slow thing.
Well,
Or
so the induction process, I mean, again, slow compare, it
yeah,
depends on what time scale.
mm,
The induction time where they really studied this, separated out induction versus maintenance,
mm,
is only about is less than 20 minutes.
okay, yeah,
Okay.
yeah.
But you may not see the expression of the plasticity for longer.
Mm. Oh, okay, exactly. So the exactly. So, but I think so. I mean, if you sort of study networks without synaptic plasticity, right? Then typically you often do it for a second or a couple of seconds, right? Or because then in order to do,
Really?
well, I think if you do these large scale models of like large networks of like some thousands of neurons, then often you don't have the capacity to run them. Certainly not for 20 minutes. Uh,
Well, that's interesting because I thought with large networks, one of the things you wanted to do is study behavior. And how can you study behavior if you're only simulating the network for a second?
Yeah, no, I think it's well. I think often. So I mean, we talk about the same networks now, like large neural networks, right?
Right.
Yeah. So I think it's well. I think it's well. One would love to study behavior, but think often you try to compare with electrophysiological experiments or something, right? And sort of to predict sort of what you can measure maybe in the lab. Then I mean, like
Hmm.
electrophysiology and optophysiology. So then at least it's on a time scale of seconds
Okay.
because it's but I think then that's a problem with synaptic plasticity, right? That if you want to, it's a much slower process. So that if you want to simulate it, you need to run it for a longer time.
Right. Well, at least experimentally, they do sometimes, often we'll see effects within minutes
Mm,
depending on the protocol.
yeah.
But I would say one of the difficulties is not so much the simulation time because honestly, that's computer time. I can go home and let the computer work.
Yeah.
But the difficulty is there are so many different stimulation protocols. And if you even if you look at a few different labs using the exact same protocol,
Mm,
you'll notice the age of the animal might be a little different or the actual solutions might be a little different.
mm,
And some of these could have an effect on the outcome and some don't. And so you have some labs which do what you think is the same thing and get opposite results, and then as a computational scientist you're left with trying to figure out what is the source of the difference. And that's why collaborating with experimentalists is quite valuable because or in my lab we also do experiments.
yeah.
You get a nice feel for all these things which can change the outcomes.
Mm,
So the difficulty is trying to match a large body of data,
mm.
which itself may not be consistent.
But is there some kind of sort of I know with for example, when it comes to like this electrophysiology, optophysiology in like let's say mouse visual cortex.
Hmm.
The Allen Brain Institute has done this wonderful work of sort of doing these same experiments for many mice, and so that you sort of actually have
Oh,
to make
that's
this as
nice.
yeah, that's nice. But and also there is this what's called International Brain Laboratory, right? Where I'm not quite, but I think there's sort of the many labs who have sort of collaborated or decided to do experiments, essentially decided on the same protocol, so that it's a little bit easier to and so that you sort of essentially pool data of the same type. But is there some kind of a collaboration within the signaling pathway that you could sort of agree on some protocols, or is it sort of every
Well,
man or woman to himself?
are you talking about the experiments or the modeling aspect?
Experiments.
So I am not as familiar with that experimental community as, say, with the electrophysiology.
Mm,
Um, and I think in terms of the signaling pathway, um, it's really not those biochemical reactions which are critical for us to understand the dynamics, but it's really trying to reproduce the synaptic plasticity data with the signaling pathway data. So I'm not really sure it's those protocols that are the issues in
I
this
see.
in the systems biology, though it might be for that
Yeah,
community.
yeah.
But
I
I
see.
feel that because we're bridging it, we're going to the systems biology community to get the data to then explain the electrophysiology.
Mm. So
Um,
electrophysiology for exactly for the synaptic plasticity.
right,
Yeah,
right.
yeah. So what kind
And
of experiments? Yeah, sorry.
those are the protocols that can be just a little different from lab to
Mm,
lab, or what what I think is an issue is that
mm.
they often only use males,
Male
and
mice.
yes,
Oh, yeah. I see.
or a particular age, which is developmental. And
Mm.
what we want to know is what happens in adults. So that's
Ah,
other issues.
I see. Because it's experimentally simpler, maybe to with the
No,
mice, or it's just
well, let's just say that some people will say, "Oh, the males and females are the same, so
mm.
we don't need to use females." To which I would reply, "If they're
What?
the same, why not use the females?"
Uh,
And then they'll say, "Oh no, the females are too variable, so we can't use them." To which I
oh,
would reply, "Oh, so they differ, so we need to use females." So in my own lab, actually, we started doing a theta burst protocol in the striatum, which is a part
mm.
of the basal ganglia, which is not the cortex. And
So
we
that's a
were getting
protocol for electric for stimulation.
right. That is it's a bit closer to what's happening in a real brain than, say, the typical 100 hertz. And
Yeah.
we were getting LTP and then it disappeared. And it was because it was the females that weren't exhibiting LTP. And then we tried to check the estrus status without stressing them out too much, and that wasn't working. So
Mm,
we ourselves are guilty of having a few publications where we only used male
mm,
mice. But then I had a student and I said, look, let's check estrus. Let's use vaginal cytology. That's the gold standard, but let's do it post mortem so that we don't treat the males or females differently. And what we discovered is that the female mice in the low estrogen stage of their cycle did indeed exhibit LTP, just like the males.
mm.
But the females who were in estrus, meaning high estradiol, did not
What does that
exhibit.
mean? That they're ovulating, or what does that mean? Or yeah.
Yes.
So that's part of their menstrual cycle.
Their estrous cycle. So only
Okay.
primates have menstrual cycles and all other mammals have what's called estrous cycles.
Okay.
But
Mm,
it's a similar concept in that the hormone levels are fluctuating and only at one time during the cycle eh, is the egg released and therefore
oh. So
will
there's okay. So yeah. So here, of course, we can start to speculate whether there are certain periods where the females are better able to learn new things, or I mean, a female mice.
yes. Well, in this case, it would be the type of learning, and there has
Mm,
been some behavior. So the striatum is responsible for goal directed learning and habit learning.
mm,
And so there are certain periods during their estrous cycle where they're more likely to use a more spatial or hippocampal based strategy.
I
And
see.
indeed, that is during the high estradiol state. So
Oh,
it could be that they actually it's not that they don't learn as well, although that's possible. It could be they use a different strategy to learn or use a
huh,
different part of their brain. And this may have implications for addiction because even human females, they become addicted faster. Once they start
huh,
using, they become addicted faster than males. And there are definitely periods during the menstrual cycle where they are more likely to relapse.
oh, wow. Interesting. So, okay, so now, but when you make models, and I think so, so most of your, or at least when you make models with these large sets of ordinary differential equations,
Uh huh.
for this reaction, or for this what's called mass action, or yeah, mass action dynamics. That's what we call mass action dynamics. So how do you? I mean, then you have a bunch of parameters, right? You typically have to assume something. Well, you have to assume something about the initial conditions in terms of the concentrations of all these molecules, and then you have to assume something about the rate constants and these equilibrium constants for all these chemical reactions,
Right.
right? So it's a bunch of
Well,
parameters, or
I don't actually like saying we're assuming something.
okay,
But as you say, we also assume channel densities
yeah,
and those types of models. No, instead, I like to think that we spend a long time reading the biochemical literature trying to find measurements of the kinetics.
mm,
And for some of the molecules, we have those measurements, so we use those. In some cases, we'll also have concentration measurements, and we'll use those.
mm,
And then when we don't have those rate constants, we have other data of not single biochemical reactions, but as you mentioned, the live cell imaging data that we can use along with the parameter optimization algorithm to
yeah,
tune the parameters to match the data.
mm. Huh. I see. So there's like a, I mean, in principle, these rate constants, I mean, that has to do with sort of, or I mean, how what happens when two molecules come close to each other, right? And
Right.
that is something that you, in principle, should be able to compute from like quantum mechanical
So if we had the
calculations.
activation energy, we
Okay.
could figure that out, but activation energy for most of these reactions isn't known. I
No.
remember searching for that also.
Huh.
Um,
So it's one of these things that, in principle, should be possible, but maybe not in practice.
so I guess there's another field of computational biology called like which does molecular dynamic
Yeah.
simulations,
Mm.
right? And they've begun to be able to do two molecules at a time.
Okay.
And I've talked to people who do that, and they said they're not yet at the stage where they can actually figure out rate constants
Ah, I
because
see.
that would be very nice.
Yeah.
I mean, the probability of two molecules reacting, right, depends on what is how fast are they colliding, right? What is the
Mm.
force of their collision? But
Mm.
also, what is the conformation of the molecule,
Yeah.
right? Are
Mm.
they colliding in the right
Yeah. Huh.
orientation?
Yeah.
Um, and then, if they do collide with enough energy and in the right orientation, then I guess that's where um, maybe your other physical principles can figure out
Mm. Yeah. The number. Yeah. At least
the
sort of it.
numbers. But
Yeah.
there's so many, yeah,
Yeah.
there's so many issues there.
It's, yes, it's very hard still to do this kind of, yeah. Even though we know the Schrödinger equation, and it's still hard to use them in many of the situations you're interested in.
Right.
Mm. So uh, okay, so you have all these parameters then that you sort of compile. Is there some are there some databases where these things
There
are
are.
put together
Um,
so that new researchers can benefit from that?
yeah, so Upinder Bala at
Mm.
NCBS in Bangalore, India, has a database where he tabulates all of the reactions and rate constants that he's used in his
Oh,
models. And
excellent.
that's a really nice resource.
Yeah.
There are some other resources in general in the systems biology field, databases. One is called Brenda. Um, I think that's run out of Switzerland. That has tabulated rate constants for many, many species, including bacteria, and they give you the conditions, um,
Mm.
but they're not those. I think you're more likely to find them, say, in the metabolic pathways. Like if you're interested in cofilin and those reactions, that's
Mm.
harder to find those rate constants in
Yeah.
those
Mm.
databases.
So is this like a community thing where people can contribute new rate constants and or is it more like
Yeah, I think it's curated, right?
mm,
The
and
rate
like this?
constants come with the
Yeah,
publications.
yeah,
And
because there
then
are
Wiki Pathways doesn't have the rate constants, but there's lots of different pathways there, which I
yeah,
think is also garnered from the literature.
yeah.
Um,
Cool. Yeah, that
but
seems
in
to be a nice important community thing
Yeah,
to try to build up.
and then there's some databases, just like in computational neuroscience, which has models, so biomodels database.
Mm.
And so the systems biology community has a standard model specification language called SBML. So I think, and so that allows them to exchange models and run it on different simulators.
Mm.
And I think biomodels might have those only SBML models, but I wouldn't,
Mm.
I'm not certain of that.
Okay, so now we talked about sort of like dynamic, well mixed solutions where the basic unit is concentrations, but and then you end up with like deterministic, like the normal differential equations, like deterministic,
Ordinary differential
ordinary, yeah,
equations. I wouldn't call
yeah,
them
but
normal.
no, no, but I think also ordinary, but also without the noise term or anything stochastic in them.
Right,
Yeah, so
right.
that's so they're okay. So, but this was a new term. So ordinary without any stochastic thing. But now, in some cases, you have to use stochastic approaches.
Yeah, well, you should use stochastic
Yeah.
approaches. If you're interested in what's going on in a single spine,
Mhm.
one molecule has a concentration of 17 nanomolar.
Mhm.
And so clearly you cannot have a concentration
So
of
that's
20,
that. This is like calcium, or what? Or you
any
said one
molecule,
animal
right?
molecule.
If you have a spine, it has a certain volume,
Yeah.
right? So
Okay.
the concentration
I see.
is,
Mm.
right, the number of molecules converted into moles,
Okay.
right, with
So
Avogadro's number,
okay. So
and you
one
get a
molecule in this
concentration.
spine volume is corresponds to the 17 nanomolar.
Yes.
Mm.
And so mean concentration of calcium in the spine is about, let's say, 60
Yeah.
or 80 nanomolar.
Uh,
So, well, it obviously is not exactly 60
no.
because 60 nanomolar is not evenly divisible by 17.
No.
So,
Exactly.
so maybe it's
Exactly.
54,
I see.
or I'm
I
not
see.
good at multiplying by
No.
17.
No.
Um,
That's true. So, but it's like three, whatever, three, four, five calcium
Yeah,
molecules in there in
right.
the
And
spine.
so let's say you have four calcium molecules and
Mm.
you have five proteins it can bind to. So
Ah.
an ordinary differential equation solving this simultaneously might say, well, a half a molecule is bound
Mm.
to
Uh,
one protein and
yeah.
two-thirds of a molecule is bound to another protein. It obviously doesn't say that in units
No.
of molecules. It tells you in concentration, but
Mm.
that's what it translates into.
Yeah.
And so you should really be doing things stochastically. What
Mm.
that means is probabilistically. You're saying at this time step, what is the likelihood of calcium binding each of its partners?
Mm.
And then you flip a coin and you choose which of those partners it's actually going to bind to.
Ah.
And
Mm.
that's also called Monte Carlo simulations because of the association of Monte Carlo with gambling,
Mm.
where you flip a coin or you pick a
Exactly.
card and see
Yeah.
what happens.
Mm. So exactly. So then you end up with like, is it like mathematically much more demanding or computationally much more demanding?
Yeah, so it is more computationally demanding because random numbers are expensive to produce computationally.
Mm,
So that would be one reason.
mm,
And then you're now back to the situation of experiments
mm,
where you get a different answer every time you run it. So you have
mm,
to run them a bunch of times.
mm. Yeah, exactly. Something like a trial average
Yes,
in some sense.
yes.
Yeah. But are there, I mean, in for doing like simulations in competition with neurons, you have all these different simulation tools which are developed, right? And Neuron and Arbor and whatever, Moose and so on. Do you also have tools which have been developed to do sort of this your kind of simulations?
Yes, absolutely. So there's many tools developed again by the systems biology community. Most of those, and some of them are very good, but
Mm-hmm.
most of them do not do spatial models. And
Mm.
then, but a few of them do. And then you have other
Okay,
models.
because we haven't talked about spatial models, but we're coming to that.
Yeah,
That's a spatial so spatial models means that you have different compartments or something that space is included, spatial variation.
exactly.
It's not well mixed. This well mixed
Right,
approximate. Yeah.
right.
Okay.
So,
Mm,
so you have these tools in the SBML community, and even aside from the space issue, most of them just model the molecules. So, if what you were interested in was how the molecules interact with the voltage of the
mm,
membrane potential, then those tools are not good. There's a couple, I think, in the system biology community, but mostly now we're turning to computational neuroscience tools, and Moose can do
mm,
um, both deterministic and stochastic signaling pathways interacting
mm.
with
Cool.
the membrane potential. And Neuron now has a module more recently added that can do
So that
some
mm.
signaling pathways.
So Moose is still developed by Upi Balas Group
Yes.
in Bangalore, right? Yeah.
Hmm.
Okay. Excellent. So okay, so then you have okay, so we have stochastic approaches if their concentrations are really really low, and then you have these spatial approaches. So
Right.
that's when. So when are they? Yeah. So when are they needed? That's they seem like a pain.
Yeah, you could say that, but when are they needed? Well, you could argue always. I mean,
Mm,
a neuron is a spatially, has dendrites, is very
mm,
elongated.
yeah.
Um, and things happen out on the dendrites. In fact, let's say you have multiple spines. I mean, for the most part, you don't get memory storage in a person when a
Mm,
single spine on a single neuron is stimulated, right? Um, you have multiple inputs representing different patterns,
mm,
and in some cases you do get memory storage, in some cases you don't. So there must be some sort of interactions among those synaptic inputs
mm,
that occurs. And so it's those interactions which are critical. And some molecules, because like calcium, because they're heavily buffered, tend not to diffuse out of the spine.
mm.
And other molecules can diffuse out of the spine. So you have these interactions that I think are critical to understanding why certain protocols produce plasticity and others do not.
Yeah, because I mean, okay, there's one candidate is that it's like the membrane potential varies in the dendrite, but I think you're saying that that is maybe not that's this idea that it's many dendrites are electrotonically compact, so the membrane potential is sort of is sort of like it's not varying at the fine enough spatial scale to maybe to be able to do this kind of
No,
fancy
I
thing?
mean it definitely is varying, but
Mm,
at a different spatial scale for sure. So if you have a 20 micron segment of dendrite,
mm,
the variation in voltage from one end to the other is very small. On the other hand, the voltage in the spine head is different than the voltage in the dendrite. And that voltage will activate calcium permeable channels and
yeah,
influence synaptic channels.
mm,
And so the calcium probably depends more on the voltage. But these GPCRs are different, right? So they're producing other second messengers. And again, a key one is cyclic AMP, and another one related to calcium is IP3. And those can diffuse a little bit further. And for example, dopamine, a critical molecule involved in a whole host of diseases such as addiction. And of course, it goes away in Parkinson's disease. And not every spine has a dopamine synapse on it.
Mm.
Maybe they're on the dendrite nearby.
Yeah.
Maybe it's a couple spines down. So if you're going to get cyclic AMP or PKA, which is critical for synaptic plasticity in the striatum, then
Yeah.
diffusion matters, and you
Mm.
have to have these interactions.
Mm. Yeah. So,
Plus, we know a signal has to get to the nucleus,
mm,
and it's not just the voltage that it's only the voltage in the soma which is involved. There's been some really elegant experiments showing that signals kinases activated in the dendrite can indeed get to the nucleus
so it
if
moves
you have
down. Yeah.
if you have
So
sufficient.
kinase is a type of protein, right?
Yes, that's
Mm.
the kinase is the molecule that puts the phosphate onto the protein,
Mm. I
and the
see.
phosphatase takes it off.
Yeah. Hmm. And but I mean, a protein to diffuse, that takes quite a bit of time, right? It's a very
Yes,
it takes must but take. I mean, at least if the neuron, I mean, if to diffuse a millimeter, that takes forever probably for a protein.
it does.
Mm.
So when they see signals showing up in the nucleus, it is, I mean, I think tens of minutes later
Okay.
and
Yeah.
at very low concentration.
Huh.
And so I'm not saying that the signal, the depolarization of the soma, I'm not saying that's irrelevant,
Mm.
but apparently, and not only that, but the proteins, here's another key question. The 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 genes are transcribed in the soma, right?
Mm.
And then either the transcript, meaning the mRNA, or the protein synthesized in the soma has to get back to the spine,
Yeah.
right?
Yeah.
That also,
Yeah.
there's almost
Yeah.
no way around that.
No. I
So
guess
there
you
are
have this.
some
Mm.
transcripts in the dendrites, and so some of the models look at um, the activated synaptic activation leading to initiating protein synthesis in the dendrite. Then you don't have to worry about the nucleus.
Yeah. But I guess also there are. Don't you have this like this railway system that you can do some active transport of things with
Yes, that's
actins
right.
or whatever. So I don't know if they what they can put in their backpack if it can take a whole protein, but
Right. Well, and it can, I think, take a whole protein. I think that ion channels are packaged into vesicles, which are transported, and
yeah,
it goes both ways.
yeah,
So even though I said we talked about it diffusing to the soma, that doesn't mean it's actually diffusion.
no,
It
it's moving.
could
Yeah.
be packaged
Mm.
and taken to the soma.
So when you do, I mean, when you model these things, because I know you do well, uh, the details of signaling along the dendrites and involving spines. Can you use this? I mean, I guess this reaction diffusion equations,
Hmm.
which has been, I think, I mean, I remember Turing used them to sort of to look at pattern generation and in like whatever on whatever like the skin of tigers and stuff. And
Yeah.
but and so they are continuous. Can you use do you use continuous versions or like ordinary differential equations, or do you have to mainly do this stochastic things?
Mostly we do stochastic.
Mm.
It turns out once you add in diffusion, on the one hand, things become even more complicated, i.e., slower
Mm.
computationally, but we have some very efficient stochastic reaction diffusion algorithms
Mm.
that actually are faster than doing it deterministically.
Yeah, sounds good.
Um,
So you're doing it on normal computers in your lab, or
we
yeah?
are. We
Mm.
haven't yet parallelized it. It
Mm.
does take like 8 hours for a
Mm.
simulation, so
Yeah,
we just parallelize it by running lots of different conditions
yeah,
or different
yeah,
parameter
yeah,
combinations.
yeah. Mm.
Um,
Embarrassingly, isn't that embarrassingly simple parallelization when you just run it?
yes,
Yeah, but it's very
exactly.
yeah, but that's often typically the case, right? The limitation you want to run the same model many times with different
Right,
parameters. Yeah,
right,
that's typically the way
exactly.
to do it. So this is cool. And then I mean, one thing that sort of is maybe surprising if you sort of come like many people do, come from start computational neuroscience modeling Hodgkin Huxley kind of like dynamics in neurons. And then you look at this ion concentrations and you have like more sodium on the outside than on the inside. But still, we're talking about millimolars. And the same with potassium, more on the inside than outside, but still millimolars on both sides and chloride. And then you have calcium, which is extremely well, it has very very low concentration on the inside, right? So it's like
Yes.
this and of course this is why it's so suitable, I guess, as to be a signaling molecule, because that any the calcium that comes in, like a few calcium ions that comes in, actually changes makes a dent into the calcium concentration on the inside, because there's so
Right.
few there from before.
Right. Although you could say that because it's a signaling molecule, that is why it is so tightly regulated.
Yeah, no, I agree with that. But it's also we could also say that like the other molecules like sodium and potassium and chloride, they are not good signaling molecules because I mean because they are sort of there's so much like any single action potential or whatever doesn't change the ion concentrations sizably over the inside on the outside. I mean you have
Well, actually, action potential is going to change the sodium concentration on the inside because remember, it's still very low on the inside.
yeah, but it's not like it's not. I mean, maybe it fires a lot, but it's not like yeah. I mean it. Yeah. So it and
And
it
let
also
me
it's true that you get potassium on the outside that can build up and cause trouble.
exactly, and
But
that's
then
one of the causes perhaps of traveling waves of epilepsy,
yeah.
right?
So but still, it's sort of it's not like it's well I don't know. At least calcium is very sensitive.
Calcium
Getting
is
a
very sensitive.
yeah.
Yes,
So
we can
that's
certainly agree on that.
yeah.
But
So
there
that's
are many buffers and pumps that control the
yeah.
calcium.
Yeah,
So
exactly. So this is interesting. How so this just all this when calcium get into the cell is like the whole battery of processes ready to regulate it,
that's right.
which is fascinating. So tell a little bit about all these processes.
Right. So there's a lot of proteins that bind calcium. So as
Mhm.
we mentioned, calmodulin, which is binds calcium but then goes on to do other things. Then there are, let's call them inert buffers. One of the molecules is calbindin. Another is parvalbumin. I find it
Mhm.
very curious that these molecules have been used to identify different classes of neurons for a long time without
Mhm.
even thinking about, well, what are the implications of a neuron having calbindin versus
Oh, yeah,
parvalbumin,
yeah, exactly.
right?
Yeah.
Um, and then there are other what's those are diffusible, though not very fast. But then
Mm.
there's what's called fixed buffers. Like when you do the experiments to measure calcium, you're flooding the cell with a diffusible calcium binding protein.
Mhm. Okay.
And as those molecules are diffusing in, the endogenous buffers are diffusing out
So endogenous, that's the ones which are there from
naturally
for
in the
yeah.
cell.
Okay.
But there have been some careful studies looking at like how long you're waiting for the diffusion and how much you're adding, and that have determined that there are immobile buffers. So even if
Mm.
you wait a really long time, they're just not diffusing out, and they're low affinity, meaning they don't bind readily to the calcium, but they're still there binding some of the calcium, and if it goes very high, they're ready to bind calcium.
Huh.
Now the thing about buffers is they don't actually get rid of the it's like a sponge, right? You
Mhm.
spill water, you use a sponge, you
Mhm.
mop up the water's now in the sponge. If you put the
Mhm.
sponge down somewhere else, you're going to make that other thing wet.
Mhm.
So these buffers hold the calcium, and if you now the ones that diffuse can take the calcium to somewhere else and
Mhm.
release it, or you can get rid of the free calcium by some other means, and then these molecules will now say, oh, you can have my calcium back. So
Mm.
they're more temporary storage of the
Yeah.
calcium.
Mm.
Um, so the things that really get rid of the calcium are the pumps. So there are pumps in the plasma membrane. The one that computational neuroscientists are more familiar with is the sodium potassium ATPase.
Yeah.
That's the one that restores the gradient caused by the action potential.
Mm. Huh. Yeah.
But there's two pumps for calcium. One is the sodium calcium exchanger, which
Mm.
pumps calcium out, and the other is the plasma membrane calcium ATPase. That very long name is abbreviated PMCA. It uses ATP to pump calcium out of the cell.
Hmm. Cool. Yeah. So, and then maybe you mentioned it, but you have all this organelles on the inside, right?
Yeah, so two sources of calcium inside the cell. One is the smooth endoplasmic reticulum. And on this organelle, there are two types of calcium permeable channels.
Yeah.
One is called the IP3 receptor channel. So, this channel, if it binds IP3 and calcium, it releases calcium from the smooth endoplasmic reticulum. And the ryanodine receptor channel doesn't bind IP3, it just a small amount of calcium will trigger it. And these are really important, not just in neurons. These are
Mm.
important, especially the ryanodine receptor, for what's called excitation contraction coupling in smooth muscle, including cardiac cells.
Hmm. Yeah.
So they've been studied quite a lot
Mm.
in that sense. But in addition, the mitochondria actually have calcium in them, though most people don't model the mitochondria. I think
Mm.
the mitochondria, yeah, I'm not sure dynamically what modulates mitochondrial calcium.
Because I
So
mean,
it,
it's sort of interesting. Like calcium, usually. I mean, I never heard about people having are being low on calcium. Maybe it's just because it. I mean, it's such an seems to be like this really key ion, and but I guess you don't. Maybe the body you never run out of it. I mean, I'm thinking you should
you
drink.
do. You know
You
what the
do?
body does if its calcium is low?
Uh,
It
I don't know.
takes it from your bones.
Exactly, exactly. So that's good.
So
Exactly.
it has a huge store of
Okay,
calcium.
I see. Exactly. So that's why we yeah, exactly. So we should drink our milk or whatever.
Exactly.
Yeah. So I mean, one thing that if you sort of look at I'm just thinking that if you sort of have like a neuron that is sort of like say something like a burst or some kind of property that it's sort of how it responds like on the spiking pattern
Hmm.
responding to inputs. And then if something bad happens to an ion channel, right? Then typically these neurons sort of after some time they can do something to come back to being a burster. And so so I'm just thinking that this link between neural firing, neural spiking, and what's going on inside cell is maybe that, like because you have these calcium currents, right? Calcium ion channels, so this thing of calcium currents, is that sort of like a key link between the spiking pattern and for example, sort of like the transcription of ion channels? Is that sort of a
Actually, it could be. So there was some very old work done by Gina Trigiano and then a computational scientist whose name I
yeah,
unfortunately don't remember right
yeah.
now. But they were looking at calcium as mediating homeostatic plasticity. So if
I
the
see.
neuron
Yeah.
is firing a lot, there's a lot of calcium, and that's going to activate, say, some processes to reduce the ion channel density and return firing to its normative level. Whereas if the neuron is not firing very much, calcium is low, and therefore other processes are going to occur. So, in this sense, I guess calcium was inhibiting transcription of ion channels.
Because I read one paper on this from the Eve Martyrs group with I think the first author was Tim O'Leary. So which was about sort of like you had this sort of like different, it's almost like a set rules or learning rules for the transcription where there was like really the calcium and
Yes,
feedback from
but
the calcium that set the ion channels.
the
Yeah,
work I'm referring to is probably 20 years before that, but
yeah,
still out of Eve Marder's group.
yeah,
The
exactly.
same.
So they,
Yeah,
yeah, they have done some really cool
absolutely.
work on that. So, and I think one point, I mean, in your paper, you had one paper that, in current opinion in neurobiology last year,
Hmm.
making time and space for calcium control. And there you even talked about like nano domains of calcium.
Yes.
That is actually, this is really high spatial resolution of that, like the. Mhm. So, what was the
Yes.
story there?
So, calcium again, really interesting molecule. And we already said it's required for synaptic plasticity. But what's really something of a paradox is that you need calcium for both synaptic strengthening,
Mhm.
right, LTP, and synaptic weakening, LTD. And there's been some really nice theoretical studies, one of them by John Rubin, showing that you cannot just look at the amplitude of calcium. Because if you just look at amplitude, like a trial-based model, the amplitude of the calcium during one trial, not even trial-based, but look at the amplitude of calcium using a momentary rule. So the calcium goes up really high, but as it goes down, it passes through lower calcium regions, which then now is going to translate into long-term depression. Or alternatively, if you look at spike timing dependent plasticity, um, and you adjust your thresholds to get LTD for negative timing and LTP for short positive timing, there's always going to be a long forward, positive interval where you get LTD, which typically
Mhm.
is not seen. And he did some really nice theoretical work showing this doesn't work. So, really, you have to look at other characteristics of the timing. And there's two other characteristics. One of which is, again, the dynamic, the duration. How long is the calcium above a particular threshold?
Yeah.
And this feeds into the signaling pathways because we know that certain calcium binding proteins can get activated very quickly and other ones take longer to activate, just have slower dynamics. But a second way to solve this dilemma is to not look at the concentration, but the global concentration. And by global, I mean even the concentration of the entire spine, but to look at, again, so nanodomains. Where is the calcium coming in? And is that channel, let's say you've got a calcium permeable channel
Mhm.
right next to a calcium dependent channel. So the calcium
Mhm.
comes in, it looks around and it says, oh, there's a channel here. I'm going to bind here. It doesn't even
Mhm.
see the buffers. It just goes right next door
Yeah.
and activates that potassium channel.
Yeah.
But there might be another channel across the room, i.e., across the spine, and a
Mhm.
calcium ion comes in and it looks across the room and says, oh, I want to go head over there. But before it
Mhm.
gets there, it gets snatched up by a buffer. So that's what I mean by nanodomains, that the that when you have a calcium source located super close to something that binds calcium, we're
Mhm.
going to have preferential binding there. And this is
Mhm.
not a new concept. Um, in the computational modeling field. I mean, because people have been talking about this for a while. For example, um, well, experimentally, what they talk about, we can block this calcium effect with EGTA, which is a slow buffer,
Yeah.
or we can't block it with this slow buffer, but we can block it with this fast buffer, meaning the slow buffer, the calcium has time to look around and find things that are across the room. With the fast buffer, it can't. And so they've used these different buffers at the presynaptic terminal to figure out how close does the calcium channel have to be to the vesicle release site to account for what they have observed in terms of vesicle release. So again, this concept on the presynaptic side, for sure, is like 20 years old or something, the
Mhm.
nanodomains.
Yeah.
But in computational neuroscience, for the most part, we haven't really modeled this except for a
Mhm.
few
Yeah.
studies
Hmm.
showing
So it,
how
yeah.
one
Mhm.
one particular type of calcium permeable channel will activate one particular type of calcium dependent potassium channel.
Mhm. So it really seems like there's a lot of sort of like a lot of, I mean, it has a lot of information, potential information processing in
Yes.
this kind of with calcium and all these different processes it's involved with, and calcium activated channels and yeah.
Yes,
Mm,
definitely. I mean, and that could be why calcium flowing in through NMDA receptor channels might activate LTP because the calcium binding partners are for that are there, but calcium influx through a different type of channels leading to LTD just because of
mm,
co-localization of those different proteins with
mm.
those different channels.
So yeah, cool. So okay, we're sort of approaching the end here. So, but it seems to would you sort of for all the young, eager computational neuroscientists listening on this podcast, so you think they should all go one level below, or at least go down to the signaling pathway level and join you, and is it sort of like lots of, well, what do you think about the prospects of making sense of all this?
Well, first of all, what I'd like to say is I certainly would like more
Mm,
young computational neuroscientists to join me. But
mm,
what I really tell everyone is your modeling needs to be driven by your question.
mm. Yeah.
What is the question you're trying to address? And therefore, what level of detail do you need? And by that, I mean you want to make sure you didn't put into your model the result you're looking for. So if you want to know how can a pattern of synaptic inputs produce synaptic plasticity in a single neuron, you might need to go look beyond ion channels. But there are certainly some questions that call for net modeling large networks,
Mm,
right? Like how does a pattern of synaptic inputs lead to a particular behavior?
mm.
And then no, of course you cannot model signaling pathways or ion channels
No.
because it ends up being, at least not with today's computers. Maybe
Yeah.
tomorrow you can do that. And so I think that's one thing I think experimentalists do better at is thinking about is more hypothesis driven. And I think it's partly cultural but partly practical. Once you become expert in a technique, it's still going to take you months to collect all the data.
Mm.
Okay? So you want to think really carefully about what is the critical question.
Yeah.
If you're a modeler, it takes you months to create the model, and after that, you can run all the simulations overnight, sort of thing.
Mm. Mm.
And so people will say, I don't have to think about the question. I'll just run all these different conditions and see what I get.
Yeah.
And so, which is, not
Mm.
conducive toward really being critical about what are the key hypotheses to test
Yeah.
when you can test them all with a
Mm.
flick of a switch, so to speak.
Cool. So, well, thanks a lot, Avrama, for. I don't know if you have anything to add.
I've had a great time talking
Yeah.
with you about this data. Oh, the one thing I wanted to add is to bring back, I told you the result about estradiol.
Mm.
So
Yeah.
now what we're
Yeah.
doing is trying to figure out how does the estradiol interact with these signaling pathways that we've modeled already.
Estradiol, that's this sort of like this like not the menstrual thing, which is not the menstrual because it's
Yes,
it's a mouse.
that's the hormone
Exactly,
that's
the
the
hormone thing.
female sex hormone that
Exactly.
that goes up periodically, which
I see.
impairs
I
long-term
see. Yeah.
potentiation.
Do you have a
And so
particular yeah.
how is it interacting?
Yeah.
Are its signaling pathways
Mm.
that it's interacting with to impair LTP?
Mm.
So that's our
Yeah.
our,
Well,
our next
that
next phase of our
sounds exciting,
study.
and I guess you're also in general interested in sort of explore that. Oh, maybe that's also for like a call for action to explore a little bit more the whatever the sex differences in this research, and consider males and females, and at least as many females as males.
Absolutely. In
Yeah.
fact, NIH now has a mandate that you have to include women in clinical studies and you have to use males and females unless it's really strongly justified
Yeah.
scientifically.
Yeah.
Um, although I have to say, I'm still shocked at occasionally reading papers that I know was funded by NIH, which only used males.
Yeah.
Certain fields are worse about it than others.
Yeah.
But yeah, I think we absolutely need to study both males and
Yeah.
females.
I just read this book on use of AI in medicine. And of course, if you want to train some of these AI networks to make like prognosis or whatever, like diagnosis and so on, you need a lot of data. So you need many previous patients, and then you end up with maybe AI that only works for, well, makes it, yeah, helps in the diagnosis of males but not females, and that's sort of not,
There's a huge bias in
yeah,
medicine.
that is not
Absolutely. There's so many areas. Heart attacks are
mm,
completely different in
mm,
females. They're not recognized as much. And yes, using AIs is great, but because so much of the data is indeed the dominant one. There's also a history of have you seen those little pulse oximeters that they put on your fingers?
yeah. I've sort of, yeah, I've certainly,
Yeah, do you
well,
know they don't work with pigmented skin?
oh really, yeah,
Mhm.
wow, yeah, I didn't know
So,
that. No,
yeah, there's a huge history of
yeah,
um
yeah,
yeah, bias in medicine, and yes, the AIs are going to pick up on that.
yeah,
Absolutely.
mm,
Unfortunately, the ideal was that AI would free us of our biases, and in
yeah,
fact,
yeah,
what we're finding is that many of those AI
mm,
systems are just instantiating our
that's
biases,
right.
and that's
But
really
at
a
least it
shame.
makes it maybe more easier to be aware of our biases so that we can do something about it. Hopefully.
Hopefully,
Yeah.
yes.
Mm. Yeah. Okay. Well, thanks a lot, Avrama.
You're welcome.